For decades, scientists have grappled with a perplexing phenomenon: the disproportionate prevalence of autoimmune diseases in women. Research indicates that approximately 80% of all patients diagnosed with autoimmune disorders are female, a statistic that has long puzzled the medical community. While various explanations have been proposed over the years, recent studies have shed light on a surprising potential culprit: a molecule responsible for preventing chromosome poisoning. Autoimmune disorders occur when the body's immune system mistakenly attacks its own healthy cells. With more than 80 identified autoimmune conditions, the gender disparity is striking across the board. For instance, lupus, one of the most common autoimmune disorders, shows a 9-to-1 female-to-male ratio, while in the rarer Hashimoto's thyroiditis, an astounding 95% of patients are female. However, a crucial twist in this narrative has emerged, potentially holding the key to understanding this disparity. The increased susceptibility to autoimmune diseases isn't strictly linked to being female, but rather to having 2 or more X chromosomes. This revelation has shifted the focus of research from gender-based factors to the genetic implications of X chromosome dosage. To understand the significance of this discovery, it's essential to delve into the basics of human genetics. Typically, humans are born with 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes, XX for females, XY for males. However, variations exist. Individuals with Klinefelter syndrome, XXY, and those with trisomy X, XXX, both show an increased likelihood of developing certain autoimmune disorders. Conversely, those with Turner syndrome, characterized by a single X chromosome, X0, are less prone to these conditions despite developing typically feminine traits. This pattern strongly suggests that the dosage of the X chromosome, essentially, how much genetic information from the X chromosome is being expressed, plays a crucial role in autoimmune susceptibility. The concept of chromosome dosage is critical because for most genes, having 2 active copies, one from each parent, is necessary for normal function. However, this paradigm doesn't work for sex chromosomes, given that XY individuals inherit only 1 X chromosome. Nature has evolved a clever solution to this dilemma: X inactivation. In individuals with multiple X chromosomes, 1X, or more in the case of XXX individuals, is essentially locked down in each cell. This process is facilitated by a long RNA molecule called ZIST, X-inactive specific transcript. ZIST, along with 81 protein partners, forms a complex that wraps around the extra X chromosome, silencing it through structural changes and epigenetic modifications. For years, scientists hypothesized that leaky or inadequate X inactivation could be the root cause of the increased autoimmune susceptibility in XX individuals. The X chromosome houses the largest number of immune-related genes in the entire genome, and some of these genes have been found to evade ZIST suppression. This escape from inactivation could lead to an overexpression of immune-related genes, potentially triggering autoimmune responses. A 2020 study provided compelling evidence for this theory. Female mice with a lupus-like disease showed reduced ZIST signals in certain immune cells, coupled with an overexpression of X-linked genes. Many of these upregulated genes had previously been implicated in autoimmunity, although the exact mechanisms linking this overexpression to autoimmune disorders remain under investigation. However, a provocative new theory has emerged, challenging the notion that faulty X inactivation is the culprit. Instead, some researchers propose that the very process of X inactivation itself might be triggering autoimmune responses. A groundbreaking 2024 study created male mouse models capable of expressing ZIST, a molecule normally only found in females. When lupus-like symptoms were induced in these mice, the disease's severity was significantly worse compared to normal male mice. The study also revealed that many autoimmune patients have antibodies targeting ZIST and its associated proteins. The researchers hypothesized that when cells die and release ZIST into the bloodstream, this large molecule, with its numerous identical antibody binding sites, becomes a prime target for the immune system. Under certain conditions, this could potentially trigger a cascade of autoimmune reactions. It's important to note that this doesn't mean ZIST is the sole cause of autoimmune diseases. XY individuals who don't produce ZIST can still develop these conditions, albeit less frequently. Furthermore, Twin studies have shown that genetics alone cannot account for all cases of autoimmune disorders. Environmental factors such as diet, gut microbiome imbalances, and infections likely play significant roles alongside genetic predisposition. Nevertheless, the 2024 study represents a significant leap forward in our understanding of autoimmune disease mechanisms. This research also has promising implications for diagnostics. If the presence of ZIST antibodies proves to be a reliable marker, it could lead to easier screening methods and potentially earlier detection of autoimmune conditions. While these findings are exciting, they also underscore the complexity of autoimmune disorders and the need for continued research. The historical underrepresentation of women's health issues in medical research makes these advancements particularly significant. As we continue to explore these potential explanations, we move closer to developing more effective treatments and preventive measures for autoimmune diseases. The journey to unravel the mystery of autoimmune diseases in women exemplifies the importance of rigorous scientific inquiry and the need to challenge long-held assumptions. By focusing on the genetic intricacies of X chromosome inactivation, researchers have opened new avenues for understanding and potentially treating these debilitating conditions. As we move forward, it's crucial to approach this information with a critical eye and consider multiple perspectives. The field of autoimmune research is rapidly evolving, and new discoveries are continually reshaping our understanding. Staying informed through reliable fact-check sources is essential for both healthcare professionals and the general public. In conclusion, the disproportionate prevalence of autoimmune diseases in individuals with multiple X chromosomes is a complex issue that goes beyond simple gender differences. The role of X chromosome inactivation, particularly the ZIST molecule, offers a fascinating new perspective on the origins of these conditions. As research progresses, we may be on the cusp of significant breakthroughs in diagnosing, treating, and potentially preventing autoimmune disorders. This ongoing scientific journey not only promises to improve the lives of millions affected by these diseases, but also highlights the critical importance of diverse representation in medical research.